this session, we are going to learn some details about multi-enzyme complex, especially with regard to pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex is part of multi-enzyme. First, let us know the definition of multi-enzyme complexes. These are the stable assemblies of more than one enzyme and generally involved in sequential catalytic transformations. Let me explain. There are several copies of one or several enzymes which are packed into only one assembly. There is only one assembly, but there may be more than one enzyme, and they carry out a series or single biochemical reactions which are taking place in the cell. With that introduction, let us go to the examples of multi-enzyme complexes. They are pyruvate dehydrogenase complex, fatty acid synthase complex, glutamine synthase complex, proteasome, rubisco. To mention, these are some of the examples of what multi-enzyme complexes. Let's go to our topic, which is pyruvate dehydrogenase complex. This pyruvate dehydrogenase complex is a multi-enzyme complex, and it catalyzes the conversion of pyruvate into acetyl CoA. Let me say in this manner, pyruvate is oxidatively decarboxylated by the pyruvate dehydrogenase complex to form acetyl CoA, and this entire process, it happens in the mitochondrial matrix, not in the cytoplasm. And this reaction is irreversible reaction. You can see here, this is an irreversible reaction which is taking place in the mitochondrial matrix, and it is the link between glycolysis and citric acid cycle. What is glycolysis? The formation of pyruvate from the glucose is nothing but glycolysis. And what is citric acid cycle? It begins with the acetyl CoA. And this reaction, it is the link between the glycolysis as well as TCA cycle or citric acid cycle. And it is irreversible reaction. This multi-enzyme complex, what we are talking about, PDH, multi-enzyme complex, pyruvate dehydrogenase, is large and highly integrated complex of 3 kinds of enzymes and 5 coenzymes. Let us know what are these 3 enzymes and 5 coenzymes. The enzymes and coenzymes are pyruvate dehydrogenase complex. First, let us go to enzymes. There are only 3 enzymes what you have to remember in PDH. One is enzyme 1, enzyme 2 and enzyme 3, and they have specific names also. First one is the name itself, pyruvate dehydrogenase. Second enzyme E2, it is called as E2. Its name is dihydrolipoyl transacetylase. The third one is dihydrolipoyl dehydrogenase. Just see these, examine these names and understand, then you can very easily understand the process of the reaction. It is dehydrogenation, it is dihydrolipoyl transacetylase, it transfers, and again it is dehydrogenation reaction. Remember, there are 3 enzymes in the pyruvate dehydrogenase complex. What are they? E1, E2 and E3. Let's see what are the coenzymes which are the part of the same pyruvate dehydrogenase enzyme. There are 5 coenzymes. How many coenzymes? 5 coenzymes are there. First one is thiamine pyrophosphate. Second is lipoic acid, FAD, coenzyme A, and NAD. FAD means flavin adenine dinucleotide. What is NAD? nicotinamide adenine dinucleotide. These coenzymes, 5 coenzymes or cofactors, they function as substrate. Some act as catalytic cofactors, some act as stoichiometric cofactors. Catalytic cofactors means they act as part of the substrate. For example, TPP and lipoic acid as well as FAD, they act as catalytic cofactors. This coenzyme A and NAD plus act as stoichiometric cofactors just to balance the reaction. They are there. And let me say the enzymes are E1, E2, E3. Coenzymes are TPP, lipoic acid, FAD, coenzyme A, and NAD plus. And this pyruvate dehydrogenase complex, it is marvelous in the fashion that it holds these three enzymes E1, E2, E3, which are carrying E2 would be in the middle. Let me explain this. This is E2, which is in the middle, and E1 is present to one side, and this is E3. E1, E2, E3. And E2 plays an important role in transferring the molecules from here to E1 to E2 and E2 to E3. E2 plays an important role, which I forgot to tell before. See here, the conversion of this pyruvate into acetyl CoA. What is our reaction? The conversion of pyruvate into acetyl CoA. It is done by pyruvate dehydrogenase complex. This conversion, it consists of 3 stages. What are the 3 stages? Decarboxylation, oxidation, and transfer of acetyl groups to coenzyme A. And in this picture, you can clearly see these 3 reactions, these 3 stages. One is the decarboxylation. Pyruvate, remember it is 3 carbon. 1, 2, 3 carbons are there. Here, this is nothing but CO2. First of all, this carbon dioxide released, decarboxylation step. The remaining molecule, this molecule is nothing but acetyl group. Acetyl group first undergoes oxidation, removal of electrons, then undergoes the transfer. Yes, here transfer of what? Transfer of acetyl groups. Transfer of acetyl group to coenzyme A to form acetyl coenzyme A. Simply this is the reaction what we are going to see in detail. Listen, though there are 3 stages are there, which is this reaction is completed in 4 steps. What are these 4 steps? Decarboxylation, oxidation, formation of acetyl CoA. All these 3 stages we have seen. The last one is formation of NADH, or it is also called as regeneration of lipoamide. These are the 4 stages. The first stage is decarboxylation. Decarboxylation, you know that it is nothing but removal of carbon dioxide. And removal of carbon dioxide from which molecule? From the pyruvic acid molecule or pyruvate. This reaction is catalyzed by pyruvate dehydrogenase complex, which is called as E1, and it is the part of what? Part of the pyruvate dehydrogenase multi-enzyme complex. See here in this reaction, pyruvate is there, 3 carbon compound, 1 carbon, 2 carbon, and 3 carbon. This pyruvate first reacts with one of the coenzyme thiamine pyrophosphate. This is the thiamine pyrophosphate. You can see the phosphates, 2 phosphates attached sequentially. That is why it is called as thiamine pyrophosphate. This is thiamine and this is pyrophosphate, thiamine pyrophosphate. In the presence of this enzyme E1, it gives rise to hydroxyethyl TPP. Remember, this carbon which I am highlighting, it is the center carbon which ionizes to form carbanion. It acquires negative charge, which thereby it readily accepts this acetyl groups. Remember this molecule, these 2 carbon molecules. But while accepting this, here when it is accepting this molecule, These molecules, they came from pyruvate. Why it is called as hydroxyethyl? Because two carbons are there, CH3. CH2 is nothing but ethyl group. Because there is hydroxyl group, it is called as hydroxy. There are two carbons, that is why called as ethyl. Hydroxy ethyl, which is attached to TPP. This entire molecule is thiamine pyrophosphate. That is why it is called as hydroxy ethyl TPP. And in this reaction, this carbon dioxide molecule is released. This reaction is called decarboxylation reaction. Very easy reaction. Pyruvate in the first step undergoes decarboxylation, forms hydroxyethyl TPP. The second reaction is oxidation reaction. Oxidation means removal of hydrogen or addition of oxygen. What happens? Electrons are, and in this oxidation, it is catalyzed by the same enzyme, pyruvate dehydrogenase, even enzyme only. In this, what happens? Hydroxyethyl TPP, it reacts with lipoamide to form thiamine pyrophosphate and acetyl lipoamide. Hydroxyethyl group attached to TPP. Here hydroxyethyl group, this is the hydroxyethyl group which is attached to TPP, is oxidized to form acetyl group. Previously it was hydroxyethyl, now it is, you can see here, it is acetyl group. CH3CO is nothing but acetyl group which is transferred to lipoamide. This is lipoamide. Lipoamide, you can see here, there is a disulfide, two sulfurs, disulfide group which is attached here, which is a part of the lipoamide. And this lipoamide is derived from lipoic acid. And this lipoamide, it acts as oxidant. Oxidant means it has to oxidize. It acts on this hydroxyethyl TPP and it forms acetyl lipoamide. Oxidant is disulfide group of lipoamide which is reduced to disulfhydryl form. This reaction ultimately forms acetyl lipoamide. This is the second reaction. Hydroxyethyl TPP, from this acetyl lipoamide, acetyl group is generated. The third one is formation of acetyl CoA, which is catalyzed by the second enzyme, which is nothing but hydroxylipoyl transacetylase. Transacetylase transfers acetyl groups. Acetyl group is transferred. What is transferred here? Acetyl group is transferred from where to where? From acetyl lipoamide to coenzyme A, and forms what? It forms acetyl CoA. The same is represented here. Acetyl lipoamide, which was formed in the previous reaction, now reacts with coenzyme A in the presence of enzyme. Which enzyme? Now it is E2 enzyme. E2 enzyme, dihydrolipoyl transacetylase. It forms acetyl coenzyme A. Okay. So this E2 enzyme transfers acetyl groups to CoA, forming acetyl coenzyme A, and in this reaction dihydrolipoamide is formed. The same is represented here. This is acetyl lipoamide reacts with coenzyme A. CoAsh means this is the sulfhydryl group, and this is coenzyme A is a very big molecule, just simply represented as CoAsh. Here coenzyme A, and this disulfide group thio linkage is preserved here. Thioester linkage, it is preserved and to form acetyl coenzyme A. Our goal is to generate acetyl coenzyme A, the end product. This is the end product which is achieved in the third step itself. But here, remember what is formed here? Dihydrolipoamide. Dihydrolipoamide is what? Here, two hydrogens are attached to this lipoamide. This dihydrolipoamide, the reaction is not end here, but this dihydrolipoamide, it has to be converted into lipoamide. Why it has to be converted into lipoamide? Because this is the enzyme. See here, this is the lipoamide in the previous reaction, which is reacting in the second step. But this lipoamide, what happened to this one? It is converted into dihydrolipoamide. Dihydrolipoamide. Now, this has to be lipoamide has to be regenerated. For that one, there is the fourth reaction, which is nothing but formation of NADH, or it is also called as regeneration of lipoamide. What is this? Regeneration of lipoamide. In this one, oxidized form of lipoamide is generated. What is reduced form of lipoamide then? This is the reduced form of lipoamide. And this is the oxidized form of lipoamide. So the main motto of this reaction is to regenerate the lipoamide. From where? From dihydrolipoamide. And what is the enzyme that is used here? It is dihydrolipoyl dehydrogenase, the third enzyme, E3 enzyme. In this reaction, the two electrons are transferred to FAD. From here, two electrons are transferred to FAD from dihydrolipoamide, which transfers the enzyme E3. It transfers two electrons from dihydrolipoamide to FAD. In turn, FAD is formed. But remember, in turn, there is electrons are transferred from FADH2 to NAD+. Thereby, NADH is formed. This electron transfer to FAD is unusual. Why it is called unusual? Because usually what happens? The common role of FAD is to receive electrons from NADH. Usually it receives electrons from NADH, but what is happening here? The electrons are transferred from FADH to NADH. This is because the FAD is attached to the enzyme. Whenever it is attached to the enzyme, the reverse process is also possible. The proteins, which is the protein here, E3, dihydrolipoyl dehydrogenase, usually or else pyruvate dehydrogenase complex. These are the proteins which are associated with FAD or FMN. Whenever they are attached to these FAD or FMN, they are called as flavoproteins. Okay, the gist of this reaction is regeneration of lipoamide from dihydrolipoamide, thereby forming what? Ultimately NADH is formed. From where? From FADH to NADH is formed. Okay, this is the overall reaction of pyruvate dehydrogenase complex. What is the reaction? Here the pyruvate is converted, which is the end product of glycolysis, is converted into acetyl CoA in the presence of pyruvate dehydrogenase complex. This pyruvate dehydrogenase complex comprises of three enzymes, E1, E2 and E3. Pyruvate dehydrogenase, dihydrolipoyl transferase, dihydrolipoyl dehydrogenase, three enzymes, as well as five coenzymes. What are the five coenzymes? Thiamine, TPP, lipoic acid, coenzyme A, FAD and NAD are the five coenzyme. Acetyl CoA is the main product of pyruvate dehydrogenase complex in aerobic respiration. And this is the pictorial explanation of conversion of pyruvate into acetyl coenzyme A. First undergoes decarboxylation. This is decarboxylation. Then this acetyl group, first it is converted into hydroxyethyl, later acetyl group. Oxidation takes place. Then the transfer of acetyl group to coenzyme A, forming acetyl coenzyme A. This is the overall reaction. Let me sum up the 4 reactions. First of all, pyruvate reacts with thiamine pyrophosphate forming hydroxyethyl TPP. And in this one, it is decarboxylation. That is why CO2 is released. This takes place in the presence of E1, pyruvate dehydrogenase E1. Second reaction also takes place with the help of E1 only. In the second reaction, what happens? Hydroxyethyl TPP reacts with lipoamide, converting it into acetyl lipoamide. This acetyl lipoamide, in the third stage, it is acetyl group is transferred to coenzyme A forming acetyl CoA and dihydrolipoamide is generated. Our product, main product is acetyl coenzyme A is generated in the third step itself, but the problem is dihydrolipoamide is there, but this dihydrolipoamide has to be converted into lipoamide. Why? Because the PDH complex again has to convert one more substrate into product. That is why lipoamide has to be regenerated. For that, this is the last step. Dihydrolipoamide reacts with FAD forming lipoamide. Dihydrolipoamide is oxidized forming lipoamide and oxidant is FAD converted into FADH2. Electrons are transferred from here to here, thereby NAD plus is converted to NADH. The overall reaction is pyruvate reacts with acetyl CoA in the presence of acetyl CoA and pyruvate. The pyruvate molecule in the presence of CoA and NAD plus, all the 3 enzymes and cofactors, 5 cofactors forms acetyl coenzyme A. Decarboxylation, carbon dioxide is released and producing NADH molecule. Okay, this is the overall reaction of pyruvate dehydrogenase complex, and pyruvate dehydrogenase is nothing but the example of multi-enzyme complex. Thank